Good day, welcome back to tuition2u.com. You are now with V. Sundram and we are now in SPM Biology subject. Currently we are doing trial for the year 2020 Biology paper, trial papers for year 2022, and the paper we are discussing now is Kelantan state trial paper, and we are going to start with the first structure question. So let's dive in straight into the question. So the first question is always, normally easy question. So let's quickly answer this to save time. So diagram 1, always read the question. So diagram 1 shows the structure of an animal cell. So they have already labeled for us X, Y and Z. So they, looking at the diagram, you automatically know they are going to ask us to identify this. So the first one they ask you to identify is X and Y. Okay. So they are going to identify X and Y, but what we are going to do is, since again we are making this trial paper as a revision for us, so we'll identify as many components of the cell as possible. So the first one that we can identify is, so always the largest structure that you have inside, spherical and largest structure that we have inside the cell will be nucleus. And after the nucleus, these lines that you see inside cytoplasm, these random lines that you see inside cytoplasm, they are known as microtubules. So these microtubules are something new in the syllabus. The only place where we are going to use this microtubule is when we are explaining the structure X. The structure X is centriole. Centrioles are made of microtubules. So that's the only information in the whole of SPM syllabus. This is the only place where we are going to use this microtubule. Later cell division, centrioles controls formation of spindle fiber. The spindle fibers are also made of these microtubules. So that's the only explanation for microtubule. So we have a smooth endoplasmic reticulum. Smooth endoplasmic reticulum, basically we have to find the structure that doesn't have ribosome. The dots that you see in the cytoplasm is ribosome. The dots you see on this structure is also ribosome. This structure is the rough endoplasmic reticulum. So the smooth and rough, the only difference is whether they have or don't have ribosome. This is the mitochondrion, one of the easiest organelles we can identify. And some other structure that you need to know is this Y here is a lysosome. Basically it will be like a sphere, like a ball, and inside there will be some dots to represent enzymes. So that is the lysosome. This is the Golgi apparatus, one more organelle or one more component that you should immediately can identify if you have these structures that are arranged parallelly. In Malay we say susun secara selari. And the last component is ribosome. So these black dots that you have in the cytoplasm, so maybe this one is the ribosome. So these are some of the components that you need to know. These are some of the components that you need to know in the syllabus. So once we already know all of these components, when it comes to exam, you must know the name of the component, you must know the function of the component. So every component that you see here, you must know the function. Exam question will be about the function. So the first question here is they ask us to identify X and Y. We already identified earlier. So the X is the centriole, the Y is the lysosome. So that's considered easy, 2 marks for us. Quite easy. So any other component they ask also, you should be able to answer. But here they're asking you for centriole and lysosome. Okay. So that's 2 marks. Next question. So match the component Z to the suitable characteristic. So we go back and we see what is Z. So we know Z is Golgi Apparatus. Make sure when you're writing answer for exam, Golgi Apparatus is capital G. Small g, no marks given. So this is Golgi Apparatus. So when we see the description given in here, as I told you earlier, made up of complex arrangement of microtubules. So that is the explanation for centriole. Take note that earlier they already mentioned, they asked us to identify centriole, they asked us to identify lysosome. This is actually the description of centriole. So this is for centriole. Or maybe I just put it outside here. And small spherical sac enclosed in a single membrane. So that is actually the explanation for lysosome. So if you know this is for centriole, if you know this is for lysosome, then the remaining one should be for Golgi apparatus. So this is the right answer, which is consists of a stack of parallel flattened sac. The sac is missing. SAC, sac. If you look at this diagram, if I were to draw Golgi apparatus, we can draw it something like this. If I draw, this is one sac, this is another sac, this is another sac. We normally have like a 4 or 5. The next one we normally make it flattened in the middle, bulging at the side, and maybe one more that looks like this. So this is what we call as sac. This is one sac. This is one sac. This is another sac, and it continues. And you also realize that the sacs are parallel. So you can see they are arranged parallel to one another. So that is the description that you have in here. So Golgi apparatus consists of a stack of parallel flattened sac. The word missing here is SAC, parallel flattened sac. Okay, so that is the description of the component. It is mentioned in the textbook. If you see the word microtubule, you should automatically think of centriole. Small spherical sac is like, if they contain enzyme, it must be lysosome. Anyway, the clue for these two is actually the question earlier. They already asked us centriole lysosome. So these two is for centriole lysosome. The remaining must be for Golgi. The next question. Component X fails to function, causing spindle fibers not to form during meiosis. So state the effect of the behavior during anaphase 1. So you see there is relationship. Earlier they asked us to identify centriole. Then they asked us, they mentioned that it's made out of complex arrangement of microtubule. That is for centriole. Now they have a question about spindle fiber. So I'm not sure whether you realize. So centriole is related to the function of spindle fiber. You should already know, if you already watch my videos for Sekolah Berasrama Penuh trial paper, there is the structure question number 6. I think there is a question about cell division. In the question for cell division, I already told you, if there is a cell division question, prophase 1, metaphase 1, anaphase 1, telophase 1. I don't want to repeat because question 6, Sekolah Berasrama Penuh, I've already explained to you. That is the question for exam. I'm not simply making statement. Let's see whether you can use that again. Prophase 1, metaphase 1, anaphase 1, anaphase 2. So in here, they mention spindle fibers are not functioning. So what are the stages you need to know? These are the stages that you need to know in terms of cell division. If it's meiosis, what happens in prophase 1, metaphase 1, anaphase 1, anaphase 2? What happens in anaphase 1 is homologous chromosomes segregate to opposing poles. This is what you learn in class. Now the spindle fibers are not functioning. If the centriole doesn't function, spindle fiber doesn't function. The homologous chromosome pairs cannot segregate, cannot separate. You can just use the word separate. Cannot separate to opposing poles. And in anaphase 2, what's supposed to happen is sister chromatid are supposed to separate. I forgot to write here. Maybe I can just type it out again. So anaphase 2, what's supposed to happen is, in here what's supposed to happen, what's supposed to happen in anaphase 2 is sister chromatid separates to opposing poles. So anaphase 1, homologous chromosome separates to opposing poles. Anaphase 2, sister chromatid separate to opposing poles. I already used this answer in Sekolah Berasrama Penuh trial paper. Now we are doing Kelantan trial paper, and again we are using the same explanation. This is antonym. So if the spindle fiber doesn't form, you just use this answer, mention it doesn't happen. Homologous chromosome does not separate to opposing poles. Correct. Or you can mention the word anaphase 1. In anaphase 1, homologous chromosomes doesn't separate to opposing poles. You already know the answer. If you already do Sekolah Berasrama Penuh, no excuse. This is same answer. The answer I give you in Sekolah Berasrama Penuh, we are using it again in here. So this is bonus question. So this is 1 mark. After spindle fiber, so you see here, the question here they ask you about centriole. In here they describe about the centriole, and here they ask what happens if the centriole doesn't function. Centriole doesn't function, spindle fiber doesn't function. So if you go back here, now there is lysosome. So the next question, they already asked us about centriole. So the next question should be something related to lysosome. So let's look at the next question. So the next question mentions that component Y is usually found in a leukocyte cell. Component Y is usually found in a leukocyte. So we already know from our explanation earlier that component Y is lysosome and leukocyte is actually a white blood cell. The next sentence they mention is explain how Y component assists leukocyte to perform its function in phagocytosis. So clearly it's mentioned the process is phagocytosis, but it's only worth 2 marks. So how does the lysosome functions during phagocytosis of a white blood cell? That a white blood cell does. So I've already mentioned in the previous trial paper, which is Sekolah Berasrama Penuh, because of the pandemic that we have around us, maybe the chapter 11 related to body defense system is important. So in the Sekolah Berasrama Penuh, the essay question, the whole question was about the third line of defense. Immune response, third line of defense, types of immunity. I hope you already watched the video. Now in this question they are asking us about phagocytosis that a white blood cell does. Phagocytosis is part of second line of defense. So under the second line of defense, as I already explained earlier, we have 3 explanations: phagocytosis, fever, inflammation. The fever is actually helping phagocytosis. Inflammation also helping phagocytosis. So under the second line of defense, the phagocytosis potentially can give you 5 marks. Here we have only 2 marks. I'll give you because we are doing revision. We are answering the trial paper. We are also doing revision. So you need to know second line of defense. So what do you have in the second line of defense is this diagram here. This diagram here. Okay, so this diagram is what you have in your textbook in terms of how phagocytosis occurs. So let's go point by point. So the first point that you need to know is phagocyte. The phagocyte is the white blood cell. So extends pseudopodium towards the bacteria, envelops the bacteria. So this is supposed to be the pseudopodium. This structure here and this structure here, they are surrounding the bacteria. So the word envelope sounds a bit complex, so you can just use the word surrounds. Surrounds is also okay. So the white blood cell extends the pseudopodium to surround the bacteria. Surround. Trap also can. To trap the bacteria, to surround the bacteria, or what you have here, envelope the bacteria. Let's use some simpler word. Surround the bacteria, trap the bacteria. That's your first point. So pseudopodium of the white blood cell surrounds the bacteria. That's 1 mark. So your second point, once they surround the bacteria will be trapped. They are trapped inside what we call as phagosome. That will be your second point. Through phagocytosis process, the bacteria is trapped in phagosome. It's trapped in phagosome. Second point. Third point, lysosome in the cell contains lysosome. You can see the name of the enzyme here, lysosome. Lysosome enzyme, when you combine it becomes lysosome. So informing the examiner lysosome contains lysosome should give you 1 mark. Now what is lysosome and phagosome going to do? They are going to fuse. They are going to combine to produce something called as phagolysosome. So lysosome or lysosome is here also. Lysosome combines with or fuses with phagosome to form phagolysosome. I'm not exactly answering the question earlier. Some of the points here we're going to use. I'm doing revision of second line of defense. This might be important for your exam. So lysosome fuses with phagosome to form phagolysosome. The main point here is the next point. Lysosome in the phagolysosome destroys the bacteria, hydrolyze the bacteria, digest the bacteria. All of that words you can use. Digest, hydrolyze, destroy. Can use. That's your 5 marks. After it's digested, the digested bacteria will be released out of the cell. That's another one sentence here. But the moment you already destroy the bacteria, meaning you already protect your body. So this is worth 5 marks. If let's say the exam question is essay question. Explain how white blood cell do phagocytosis. This is the answer that you need. If you count back how many points you have, Pseudopodium surrounds the bacteria is 1 mark. The bacteria is trapped in phagosome, 1 mark. Lysosome contains lysosome, 1 mark. Lysosome fuses with phagosome to form phagolysosome, 1 mark. And the main point, which is the lysosome destroys the bacteria, digests the bacteria, hydrolyze the bacteria, is worth another 1 mark. So that's 1, 2, 3, 4, 5. That's the 5 marks. If you want to continue, the digested bacteria is expelled, released out of the cell through plasma membrane. That's worth another 1 mark. But by this sentence number 5, you already have your 5 marks. That's the main answer. How do we destroy the bacteria? Through phagocytosis. From this answer here, for the question we are answering now, you can provide this answer. Lysosome contains lysosome. The lysosome destroys the bacteria in phagolysosome. That should be the main answer. So I'm explaining phagocytosis. From this answer, we choose 2 points. What lysosome have, what lysosome do. That should give you the 2 marks. That's the main point. So let's go back to this question. So explain how lysosome assists leukocyte. Lysosome contains lysosome. 1 mark. Lysosome hydrolyze or break down or digest bacteria in phagolysosome into simple molecule. Or what we can do is, we can also add this one to relate to what I explained. Lysosome fuses with phagosome in cell to form phagolysosome. Lysosome break down bacteria in phagolysosome into simple molecule. That is the 2 marks. I'm giving you 3 sentences. The first sentence and this sentence should be enough to get you the 3 marks. So this is question 1. So question 1 is about components of the cell. The famous components of the cell is Golgi Apparatus, famous, lysosome, famous, and rough endoplasmic reticulum. And actually, Golgi Apparatus, ribosome, and rough endoplasmic reticulum, these 3 always come together. Most of the time they come together. Normally the question for Golgi Apparatus, ribosome, and rough endoplasmic reticulum is something to do with extracellular enzyme, quite famous for exam. So lysosome, Golgi Apparatus, ribosome, rough endoplasmic reticulum, Mitochondrion. These are the famous ones. So but in here they ask about centriole. Centriole is not that famous for exam, but never mind, we have already covered it. The one I've shaded is the one that normally comes out for exam. The more important components of the cell. So and normally like lysosome, they are standalone. They come out on their own. They are needed for digestion of bacteria. But Golgi, ribosome, rough, they come together for synthesis of extracellular enzyme. If the question comes out, just write back the function. Ribosome function, rough endoplasmic reticulum function, Golgi apparatus function. Mitochondrion is obviously for synthesis of energy. Energy for the sperm to swim towards the ovum, energy for the muscles to contract. Energy is produced through oxidation of glucose during cellular respiration. So that is components of the cell. So this is the first question, and the first question is giving us 2, 4, 2. So first question is always 6 marks, and by right this 6 marks is a bonus question if you have done your revision. I'll see you in the second question.